So all of last week, all we talked about was Mendel and regular mono and dihybrid crosses where nothing weird happened. And this week, we're going to talk about what happens beyond Mendel's crosses, because obviously everything is not going to be quite as simple as the pea plants that he studied. So the thing we'll talk about today is the chromosome theory of heredity. So there's a guy, his name is Walter Sutton, and in science, it's usually the person's last name that you need to remember so that you could refer to them. This was after Mendel had died, so he did all of his research, all of this work about pea plants, gave us tons of insight into genetics. And then Walter Sutton was studying grasshoppers. So maybe as a side note, sometimes people are like, oh, being a scientist, that would be so boring. No, this dude just looked at grasshoppers all day. That was his job. Some of you might remember, if you had me for Bio30E reading an article about people who studied how to pee on a urinal, that was their job. Science can be really cool. You just have to find something that nobody has studied before. So Walter Sutton found a couple of things. He found that gametes, so we're still talking about gametes, even though it's not chapter 16 anymore. Gametes follow Mendel's model. So, independent assortment, segregation. Gametes are going to get half of the genetic information that the parents have. And he also found that genes are on chromosomes. So that's fantastic. So far, so good. Nothing weird there. But he noticed something. He noticed that if two genes are on the same chromosome, the second law doesn't apply. So if two genes are on the same chromosome, the second law, so Mendel's second law, independent assortment, doesn't apply. Basically, he's saying if two genes are on the same chromosome, they get inherited together. One of them definitely affects the other one. So when two genes are on the same chromosome, we refer to them as linked genes, because you're going to inherit them together. And that is how things work in the real world. You have 23 chromosomes, you have 2 copies of each. Each of them has several genes on it, which means that you're going to inherit those together. Linked genes can separate, however, if crossing over occurs. And so I'm going to remind you all what crossing over is and when it might happen. There's a lovely little picture here of a chromosome with its sister chromatid. So that goes back to the very first chapter. If I was in meiosis, not only would I have one chromosome with its sister chromatid attached, I would also have the other homologous chromosome lining up next to it. So these two things are called homologous chromosomes. We'll call them both chromosome number 1. You have 2 copies of chromosome number 1. And if your cells have gone through interphase, you will have replicated each of your chromosomes to make the chromatid that is attached. Now, homologous chromosomes don't always line up. Does anybody remember when you would see a group of 4 like that lined up together? Mitosis or meiosis, which phase, anything like that? Meiosis, absolutely. So this thing right here, all of them, is called a tetrad. We see this in meiosis, and specifically, you mentioned metaphase. In prophase sometimes or metaphase 1, we should have homologous chromosomes next to each other, and that is when crossing over can happen. Crossing over would be if the end of that leg and the end of that one crossed over each other and exchanged some genetic information. So even though two chromosomes are on the same gene and should get inherited together, crossing over will sometimes cause them to separate. So if I was a person who makes diploma exam questions, I would make a question that was about tetrads or meiosis together with a question about linked genes because both of them have to do with the same principle. The other thing that's important to know is that crossing over is more likely on genes at the ends. So it is less likely for genes that are close to each other or in the middle to cross over than it is for genes at the ends of each of the chromosomes. So with this information, we could perform a task. We could figure out what is the order of a bunch of genes on a chromosome, and that will be our goal. Figuring out the order that genes go in on a chromosome based on how much crossing over has happened. So this is the theory that goes behind it. Does anybody want to ask questions maybe about meiosis or about crossing over? So the actual process is called chromosome mapping, and this is the skill that you will need to be able to perform. There is regularly a question about this on the diploma exam. In chromosome mapping, we're trying to determine the relative order of genes on a chromosome. And I underlined the word relative because generally the thing that we're going to ask you is which one comes first, which one comes second, which one comes third. Now, when we map a chromosome, we are not going to use actual measurements of distance because those measurements would be ridiculously small and they would be hard to work with. They would be weird decimal numbers or things measured in things like nanometers or picometers, very small numbers. So instead, because this is what scientists do, we just made up a new unit that we're going to use. We're going to use what's called a map unit. One map unit represents 1%. And that 1% is talking about the crossover frequency, which we will call recombination. So every time two genes cross over 1%, we would say that they are one map unit apart. If they cross over 50% of the time, we would say that they are 50 map units apart. So I drew, and I really did draw this, I didn't just copy someone else's picture. I drew a little imaginary chromosome. And on this chromosome, there are 3 genes that we're going to discuss. Now, my example is based on the fruit fly Drosophila, which is often used in studies of genetics. And so this is not made up. This is real, actual data. For Drosophila, on one of their chromosomes, there is a gene for eyes, for body, and for wings, all present on the same chromosome. Now, I've already showed the answer, and so I'm going to show you how I came about getting that answer. In the data, here's what we see. 10% of the time, there is recombination for eyes and wings. That means that instead of them being inherited together like they're supposed to be, they get separated. There is also 4% recombination between eyes and body, and 6% recombination between body and wings. So that is the data that I would have worked with. Now this is typically what you'll receive if it's a chromosome mapping question. You'll get a list of percentages, how much recombination has happened between these genes, and you'll be asked to place the genes on the chromosome in the correct order. So here's my strategy. Start with the biggest number. It will make your life easier. You could still get the right answer starting with some other number, but if you start with the biggest number, you can put those two genes the farthest apart. And it's very likely that the rest of the genes will be in between those two somewhere. It's not 100% for sure, but it's very probable. So you'll notice I indicated here that there were 10 units between eyes and wings. My strategy is always to go from big to small, so I would do the next biggest number. The next biggest number is 6%. I know that there's 6% recombination between body and wings. So I actually have two choices, here or way over there. I don't know which one of them is right. So generally, once you've placed the two that have the biggest distance, every time you're going to place another one, you will have two choices. Will it be on the outside or will it be on the inside? I would say 98% of the time, it will be between the two that you already had. Now, I know there's 6% between body and wings. The reason I know that body goes here is because of this 4%. I see that body has to be 4 units from eyes and 6 units from wings. If I were to put it on the other side of wings, it would be way too far away from eyes, and so the percentages wouldn't line up. So this is the general method that you will go through. There's a couple of vocabulary words that I just like to define on here because they usually get used as part of the context for these questions. Recombinant types are phenotypes from a crossover. So if we mention that this is recombinant, we mean these genes were on the same chromosome, but crossing over happened. Parental types are the phenotypes from two linked genes that are still linked. They stayed on the chromosome that they were on, no crossing over happened. And frequency of recombination means which percent of offspring showed crossing over. Now, generally, the way that this is done is that you take some organisms, you cross them, and then you collect data on their offspring. And so usually what will happen is we will have already done the cross and collected the data, and you will be asked to work with the data.